if I handed you a glass of clear liquid with a pH of -1, and I told you that your own stomach acid actually isn't too far off from that, I mean, would you drink it?
I'm going to go ahead and say probably not.
Right. Definitely not. But today, we're completely dismantling the pH scale. So whether you're a nursing student who's, pulling your hair out, prepping for the TEAS or HESI exams, or you're just an intensely curious learner, you probably think you have a pretty good handle on this stuff.
Yeah, it's one of those things we all just sort of absorbed in high school chemistry.
Exactly. It goes from 0 to 14. 7 is perfectly neutral, and it tells you exactly how strong an acid is. It feels really neat. It feels, I don't know, settled, like a universal law of nature.
It does feel that way, yeah. It has that comforting binary simplicity to it. But, well, when you actually look at the literal chemistry happening underneath the numbers, you realize it's not a rigid law of physics at all.
Which is wild to think about.
Right. It's a highly specific, very delicate framework. And the moment you change the environment, like, say, stepping from a 25-degree-Celsius chemistry lab into a 37-degree human body, all those neat little rules you memorized, they just shatter.
And that is exactly our mission for this deep dive. We're going to unpack what pH actually measures, explore how this, seemingly abstract math dictates everything from global ocean survival to the hyper-specific chemistry of human blood, and unlearn some major high school myths along the way.
Sounds like a plan.
But I have to admit, diving into this material, I got stuck almost immediately on the origin story. Because, I expected this to start with some theoretical physicist staring at a chalkboard in a sterile lab. But the trail actually starts at a brewery.
That is exactly where it starts. So the year is 1909, and we are at the Carlsberg Laboratory in Copenhagen, which was, surprisingly enough, the actual research arm of the Carlsberg Brewing Company.
That is just so funny to me.
Right, and there's this Danish biochemist named Peter Lauritz Sørensen working there. He wasn't trying to unlock the secrets of the universe at all. He was just trying to make sure the beer fermented correctly.
Okay, so what does beer fermentation actually have to do with measuring acid?
Well, it comes down to enzymes. Fermentation relies on yeast and enzymes are exquisitely sensitive to the concentration of hydrogen ions in the liquid. If the hydrogen ion concentration is off even just slightly, the yeast stops behaving, fermentation stalls, or, the flavor is completely ruined.
Oh.
So Cernan needed a way to measure and track these hydrogen ions for quality control.
But the actual problem he faced wasn't a chemistry problem, right? It was a math problem. From what I understand, writing out the raw numbers for hydrogen ion concentrations meant dealing with these comically tiny fractions.
Oh, highly prone to human error, yeah. If you're scientists doing daily quality control, writing out like 0.0000001 moles per liter in a lab notebook all day.
That's a nightmare.
It's a total nightmare. It's unwieldy. Eventually, you're going to drop a zero, misread a decimal, and ruin an entire batch of beer. So Sørensen came up with a mathematical workaround.
Oh.
He realized he could compress these tiny fractions by using negative logarithms. Instead of writing out all those zeros, he just extracted the exponent. So that massive microscopic decimal just becomes a 7.
A nice, clean, manageable number.
Exactly. And he called this shorthand pH.
I love that a cornerstone of modern science was essentially invented to save a guy from getting hand cramps while writing zeros in a notebook.
Basically, yeah.
But there's a detail here that is so funny to me. I read that nobody actually knows for sure what the P stands for. Is that true?
It's totally true. It remains genuinely disputed in the history of science.
Really?
Yeah, a lot of textbooks will tell you it stands for power of hydrogen, or maybe the Latin phrase pondus hydrogeni, meaning weight of hydrogen. But if you look at Sørensen's own papers, he was using a standard French and German notation of the time.
So it might just be a random letter.
Many historians argue the P was just a mathematical operator for an exponent, kind of arbitrarily attached to the letter H for hydrogen. It's a fitting irony, really, that this tool we use for exact, flawless measurement has an origin we just cannot completely measure.
That is amazing. And the practical innovations didn't stop with beer either. The timeline jumps to 1934, and suddenly we're talking about California lemons and a chemist named Arnold Beckman. How do we get from Danish beer math to American lemons?
Well, so Sorensen gave us the math, right? But physically measuring the hydrogen ions in a liquid was still this tedious, time-consuming chemical process.
Okay.
And the California citrus industry desperately needed a fast, reliable way to test the acidity of lemon juice for their own agricultural quality control. So Arnold Beckman invented the first practical electronic pH meter.
Okay, wait. How does an electronic meter actually measure something as tiny and, well, invisible as a hydrogen ion? I've used those little probes in biology class, but I never understood what the machine was actually doing.
It's a brilliant piece of engineering, honestly. Beckman's meter uses a very specific glass electrode. It's essentially a fragile little glass bulb filled with a solution of a known pH, and you dip it into your lemon juice.
Right.
That special glass membrane is designed to be permeable, or really interactive, only with hydrogen ions. So the hydrogen ions in the lemon juice interact with the outside of the glass, creating a tiny electrical voltage difference compared to the known solution inside the bulb.
Oh, so it's measuring electricity.
Exactly. The meter just reads that microvoltage and translates it into a pH number. And by solving this agricultural problem with lemons, Beckman essentially birthed the entire modern scientific instrumentation industry.
That is wild. And it really highlights a profound truth about science, I think. The pH scale wasn't discovered carved into a stone tablet. It was invented as an industrial shorthand. It's a tool.
A very specific tool.
Right, and like any tool, if you don't understand how it works under the hood, you're going to misuse it. Which I guess brings us to the math itself. Sorensen's brilliant shorthand giving us these simple, friendly numbers from 1 to 14 is actually mathematically deceptive, isn't it?
It is deeply deceptive, yes. And this is the single most important and probably most misunderstood property of the scale. The formal definition, and for anyone taking their nursing exams, you really need to memorize this, is that pH equals the negative base 10 logarithm of the hydrogen ion concentration.
So what does that actually mean for the listener navigating this? Because I look at a pH scale and my human brain immediately processes it linearly.
We all do.
Yeah, we are wired to assume that the step from, say, pH 6 to pH 5 is the exact same distance or amount of change as the step from 5 to 4, like inches on a ruler.
And that linear thinking will trip you up every single time because it is a logarithmic scale. Each whole number step represents a 10 times change in hydrogen ions.
10 times. So it's not a staircase. It's like an escalator that keeps accelerating.
A very steep escalator, yes. Let's look at a concrete example. If you have a solution at pH 6 and you move down to pH 3, you haven't just made it 3 units more acidic.
Wait, let me do the math. You drop from 6 to 5, that's 10 times. From 5 to 4, that's 100 times. From 4 to 3, that's 1,000 times more hydrogen ions.
Exactly. A solution at pH 3 has 1,000 times the concentration of hydrogen ions as a solution at pH 6.
That's crazy.
It is. This logarithmic compression is incredibly powerful. The physical reality of chemistry is that ion concentrations vary wildly. If you tried to map the range of hydrogen concentrations in nature on a linear scale, your piece of paper would have to stretch from the Earth to the moon.
Wow.
Logarithms allow scientists to fit an absurd range of reality onto a single, readable piece of paper. But you must remember that a seemingly small change in the number hides a massive exponential change in the actual chemistry.
Okay, so if it's just a mathematical compression trick, based on a specific set of parameters, what happens when we leave the ideal 25 degrees Celsius chemistry lab? Because you mentioned earlier that the rules start to break.
They absolutely shatter. And for anyone studying for a clinical exam, this is where you need to pay very close attention to the nuance.
Let's dismantle the biggest myth first, though, the 0 to 14 rule. We are universally taught that pH is a scale with a hard floor at 0 and a hard ceiling at 14.
Yeah, that boundary is an illusion. It is just a convenience based on the ion product of water specifically at 25 degrees Celsius. In those exact conditions, it sets a nice range where the concentrations of hydrogen and hydroxide ions behave predictably. But the math itself doesn't stop there.
Right.
If you have highly concentrated strong acids, the math drops right past 0.
How does the math even allow for a negative pH? That just sounds wrong.
Well, because of the logarithm. Remember, it's the negative log of the concentration. If your concentration of hydrogen ions is exactly 1 mole per liter, the log of 1 is 0, so the pH is 0.
Okay, tracking.
But what if your concentration is higher? What if it's 10 moles per liter? The base 10 log of 10 is 1. Make that negative, and you suddenly have a pH of -1. Highly concentrated hydrochloric acid can easily sit at a negative pH.
That is so counterintuitive.
And on the flip side, a highly concentrated strong base like sodium hydroxide can easily exceed pH 14.
Mind blown. Okay, myth number 2. Neutral is always 7. We've all seen the charts. Pure water is right in the middle at exactly 7.0.
Again, that is only true at exactly 25 degrees Celsius.
Only at room temp.
Right. Pure water self-ionizes. It naturally splits apart into hydrogen ions and hydroxide ions. At room temperature, they perfectly balance out at a concentration that equals a pH of 7. But that ionization process is temperature dependent. When you heat water up, it splits apart more easily.
On human body temperature isn't 25 degrees Celsius. It's roughly 37 degrees Celsius.
Which means the neutral point inside your own body is not 7.0. Because of the higher temperature, water ionizes slightly more, creating more hydrogen ions natively, which shifts the true mathematical neutral point down to around 6.8.
The neutral 7 fact that millions of students have rote memorized isn't even true inside the blood running through their own veins. That is staggering.
It really is. And there is a third major misconception we need to clear up too. It's the idea that a strong acid automatically has a very low pH, and that a pH number tells you how strong an acid is.
Wait, I definitely thought that. If something is a strong acid, it has a low pH. That's just, well, common sense.
It's a classic trap because strength and pH are measuring two entirely different things.
Oh, really?
Yeah, acid strength refers to a substance's chemical ability to completely dissociate or break apart when you drop it in water. Strong acids throw all their hydrogen ions into the pool immediately. Weak acids hold onto some of theirs. That is a permanent, unchanging property of the substance itself.
Okay.
But pH just measures the current concentration of hydrogen ions in the specific beaker you are holding right now.
Wait, let me see if I have this straight. You were saying I could have a strong acid, but if I dilute it with, like, a swimming pool's worth of water, its pH might actually be higher, meaning less acidic, than a weak acid that is super concentrated in a tiny test tube.
That is the exact mechanism, yes. A dilute strong acid can easily have a higher pH than a concentrated weak acid. pH tells you what is happening in the liquid right now. Strength tells you what the chemical is theoretically capable of doing.
That distinction is huge. Okay, so if the math shifts that much just from getting warmer, and a fraction of a decimal point represents a massive chemical swing, how does human biology survive it? Because human arterial blood is regulated in a window that is, honestly, terrifying.
Terrifying is the right word. Your arterial blood is tightly, ruthlessly regulated in a hyper-narrow window between pH 7.35 and 7.45.
That is barely any wiggle room. And if 6.8 is true neutral at body temperature, our blood is actually hovering in a slightly alkaline state. What happens if we step outside that window?
Well, if your blood pH drops below 6.8 or rises above 7.8, it is generally incompatible with life.
Incompatible with life.
Wow. The enzymes and proteins in your cells rely on specific electrical charges to hold their shape. If the hydrogen ion concentration changes too much, those proteins simply denature, they unravel, and stop working.
So how do we not just die every time we drink a glass of orange juice?
Because of physiological buffers.
Yeah.
For nursing students listening, this is the backbone of acid-base balance. Your body maintains this 7.35 to 7.45 window through three main mechanisms. First, the chemical buffers in the blood itself, primarily the bicarbonate buffer system.
How does that actually work mechanically? I've heard it described as a sponge, but what is physically happening?
The sponge is actually a molecule called bicarbonate. When free-floating, dangerous hydrogen ions enter your bloodstream, they don't just swim around wreaking havoc. The bicarbonate molecules physically bind to those extra protons.
They grab them.
Exactly.
Yeah.
When they combine, they form carbonic acid, which is a weak, stable acid. The bicarbonate basically neutralizes the immediate electrical threat of the free hydrogen ion.
But eventually, that sponge gets full, right? You have to wring it out.
Which brings in the lungs and the kidneys. Your respiratory system provides immediate compensation, that carbonic acid we just mentioned. It can break down into water and carbon dioxide.
CO2.
Right. So if your blood gets too acidic, your brain detects it and tells your lungs to breathe faster. This is hyperventilation. You are literally exhaling the volatile acid away in the form of CO2 gas, shifting the chemical equation in real time.
You literally breathe the acid out of your body.
You do. And the third system, which takes longer to kick in but is incredibly powerful, is renal regulation. Your kidneys provide metabolic compensation. They can physically excrete excess hydrogen ions directly into your urine, or they can manufacture and hold on to new bicarbonate to push the blood pH back up.
Okay, that makes sense.
When clinicians run arterial blood gas, or ABG, tests in a hospital, they are checking to see if these exact systems, the respiratory exhaust or the metabolic kidneys, are failing, which leads to clinical acidosis or alkalosis.
Okay, this raises a massive practical question. If our blood pH is locked into this vault, and our lungs and kidneys aggressively fight off any changes to maintain that 7.4, why is there an entire multi-million-dollar wellness industry built around the alkaline diet? People swear that eating alkaline foods like leafy greens or drinking expensive alkaline water changes their pH and detoxifies them.
The alkaline diet is a physiological myth based on a complete misunderstanding of the mechanisms we just discussed.
Yeah.
You cannot eat your way to a new blood pH.
Really, none of it works.
None of it. If a diet actually shifted your blood pH by any meaningful amount outside of that 7.35 to 7.45 range, you wouldn't feel rejuvenated. You would be in the intensive care unit with metabolic alkalosis.
But the people who do these diets buy those little test strips and they test their urine, and the pH of their urine absolutely does change. It gets more alkaline.
The urine pH shifts, yes, but that is exactly the point. The urine pH changes because the kidneys are doing their job.
Oh, I see.
Your kidneys are filtering out the excess alkaline load from that specific diet and dumping it into your bladder specifically to ensure that your blood pH does not change. Testing your urine and celebrating that your body is now alkaline is fundamentally backwards. It's just proof your kidneys are actively taking out the trash.
It's like turning up the air conditioning inside your house and then pointing to the blast of hot exhaust coming out of the AC unit in the yard as proof that your house is getting warmer.
That is a perfect analogy. The exhaust doesn't represent the house. The urine doesn't represent the blood. And just to contrast how crazy the body's compartmentalization is, while your blood is locked at 7.4, the gastric acid in your stomach sits at a brutal pH of 1.5 to 3.5.
Which is acidic enough to slowly dissolve metal.
It has to be, in order to activate the digestive enzyme pepsin and to instantly kill most pathogens you ingest. The body maintains these radically different pH zones perfectly separated.
Okay, so our bodies survive these massive exponential swings because we have kidneys and lungs constantly running damage control. But what happens to a massive biological system that absorbs acid but doesn't have an exhaust pipe to vent it all away, like the ocean?
This is where that logarithmic math we discussed earlier becomes globally critical.
Right. I read that the world's oceans have dropped from a pre-industrial average pH of roughly 8.2 down to 8.1 today. Now, to a layperson, dropping from 8.2 to 8.1 sounds like a rounding error. It sounds entirely insignificant.
But remember the escalator. pH is logarithmic. That tiny 0.1 unit shift doesn't mean a little bit more acid. Because of the math, a 0.1 drop corresponds to a massive 25 to 30% increase in the actual concentration of hydrogen ions in the water.
A 30% increase. So a decimal point change is actually a global ecological shift.
Unquestionably. As the oceans absorb excess atmospheric CO2, it forms carbonic acid, just like in our blood. But the ocean doesn't have lungs to hyperventilate it away.
Right. It's just stuck there.
Exactly. This massive influx of hydrogen ions fundamentally alters the chemistry of the water. It binds up and reduces the availability of carbonate ions. Marine life, like corals and shellfish, desperately need those free carbonate ions to build their calcium carbonate shells. It's a textbook demonstration of why you can never look at a pH number linearly. Small number change, massive chemical consequence.
It makes you realize how vital precise measurement is. Before Arnold Beckman's electronic glass electrodes, how did scientists even track this stuff?
They relied heavily on chemical indicators, like litmus paper, the classic red and blue strips.
Oh yeah, from middle school.
Right. It's a great historical stepping stone, but it has severe limitations. Litmus paper only tells you whether something is acidic or basic, not by how much. It gives you a category, not a number.
That's not very helpful for precision.
And worse, in certain environments, like with strong oxidizers or bleach, the chemical in the paper can literally be bleached white, giving a false neutral reading. That lack of precision is exactly why electronic meters became the global standard.
As we wrap up, I noticed something fascinating in the reading. We'd spent this entire time talking about pH as the measurement of hydrogen, but that's just one framework, right?
Yes, it is. The pH scale is built on the Arrhenius and Brunstad-Lowry theories of acids and bases, which define everything by how hydrogen ions' protons move around. But in 1923, a chemist named Gilbert N. Lewis expanded this definition completely.
How do you have an acid-base reaction without hydrogen?
Lewis proposed a broader framework focusing on electron pairs. Under the Lewis definition, an acid is simply a substance that accepts an electron pair and a base is a substance that donates one.
Okay, so it's about electrons, not protons.
Right. A great real-world example is boron trifluoride reacting with ammonia. There is no hydrogen ion being swapped between them, but ammonia has a lone pair of electrons, and boron trifluoride gladly accepts them, forming a new bond. It is a violent, completely valid acid-base reaction, and hydrogen isn't involved at all.
Wow.
It proves that pH is just a specific lens we use to look at the world, not the entire picture.
That completely flips the script. Okay, we have covered a massive amount of ground today. Let's quickly synthesize the takeaways, especially for those exam prep checklists. Number 1, pH is a logarithmic scale measuring hydrogen ions. Because it is the negative log base 10, every single whole number step is a tenfold or 10 times change in concentration. It is an escalator, not a ruler.
Exactly.
Number 2, 7 is only truly neutral at 25 degrees Celsius. Inside your 37-degree body, true neutral is 6.8. Number 3, your blood stubbornly guards at 7.35 to 7.45 range using the bicarbonate buffer sponge, respiratory exhaust via the lungs, and metabolic regulation via the kidneys. You cannot change your blood pH with an alkaline diet.
Please don't try.
And number 4, because of that logarithmic math, a seemingly tiny 0.1 drop in ocean pH actually represents a terrifying 30% increase in global acidity.
That covers the vital mechanics perfectly. But there's one final concept I want to leave you with to mull over. We've talked a lot today about pH measuring hydrogen ion concentration. Concentration is essentially a simple head count of the ions present. That is what you will be tested on. But modern electrochemistry actually defines pH as a measure of hydrogen ion activity.
Activity. How is an ion's activity different from its concentration?
Think of it this way. Concentration is just how many people are in a room. Activity is how those people effectively interact with each other. In a highly crowded chemical solution, ions bump into each other. They get in each other's way.
Oh, okay.
Their electrical charges interfere with one another. They socialize, for lack of a better word. The true pH is determined by that socialization, not just raw head count.
So 10 ions acting wildly might have a different effect than 10 ions that are, like, suppressed by their environment.
Precisely. So the provocative question to explore on your own is this. If the chemical reality of our world is dictated not just by how many things are present, but by how they socialize and interfere with each other in a crowded environment, how might that change the way we think about other biological or medical measurements?
Oh, that's interesting.
Are there other areas in medicine where we are obsessively just counting things when we should really be looking at how they behave?
That is a phenomenal question. It completely changes the paradigm. We started this deep dive talking about how we expect medical and chemical measurements to be clean and binary, like a perfectly neat 0 to 14 scale. But the truth is always messier, more dynamic, and honestly, a lot more interesting than the simple numbers we wrote down in high school.